Well, welcome everybody. It's just gone 5 o'clock. In fact, I think I can probably dispense with the microphone. It sounds a little bit loud for us here. I'm delighted to welcome you all today to Wolfson for our annual September Alumni Lecture and Drinks. My name is Hugh David. I'm the Development Director and a Fellow of the College. And it's a particularly great pleasure to have here today David Ray, Professor of Endocrinology, co-director of the Sir Jules Thorne Sleep and Circadian Neuroscience Institute, and governing body fellow of Wolfson. David will be speaking for about 40 minutes, followed by some time for Q&A, before we make our way to the buttery for drinks and canapes at about 6 o'clock. David began his career by training in general internal medicine in the northwest of England, and he obtained his PhD from the University of Manchester. He was a research fellow at UCLA and the Cedars-Sinai Medical Center in Los Angeles for 2 years in the 1990s, where he worked on neuroendocrine immune interaction, before returning to the UK, where he obtained a GSK fellowship to work on glucocorticoid action and sensitivity in inflammatory disease. He became a professor of medicine at the University of Manchester in 2005 and went on to study nuclear receptor and circadian biology in inflammation and energy metabolism. In 2014, he was awarded the Clinical Endocrinology Trust Medal, and 4 years later, he became Professor of Endocrinology here in Oxford. In 2020, he was awarded the Society of Endocrinology Medal in recognition of outstanding studies, and perhaps to cap it all, the same year he became a governing body fellow of Wolfson. Today, David's talk is entitled, "It's Later Than You Think, but Not Too Late to Think About Timing in Medicine."
Thank you very much indeed for the very kind invitation. It was a great honour to pass the quite rigorous interview to be allowed to become a governing body fellow here at Wolfson, and a real sort of honour to be sort of part of this community. So it's a real pleasure, to be invited to talk about some of the work we've been doing on the sort of biological timing system and its relevance for human health and medicine. So, I don't have any conflicts of interest to declare. And this is me. I wonder if this might be you. So we're currently facing an epidemic of exhaustion. So I run some endocrinology clinics up at the Churchill Hospital, and we very frequently see people referred for profound exhaustion. And it's something we don't really have a simple medical explanation for, and it's really sort of an unmet medical need, but it's hugely important for people's quality of life. So why might this be? So, sleep. So sleep's a really interesting thing. We spend a third of our lives asleep. Until fairly recently, it wasn't entirely clear what it was for, or what happened when we were asleep, and almost by definition, no one can ever remember what they were doing when they were asleep, or rather, the recollections you have, the dreams, are soon forgotten, and it's not entirely clear what they mean either. So, but here are some, I think, sort of helpful statistics about what's happening to sleep in sort of modern societies. So, actually, having a small child is a good way to be sleep deprived also, but we'll see how this pans out. So about 30% of all Americans now sleep for less than 6 hours per 24 hours. And if you're a night shift worker, it's about 44% sleep for less than 6 hours every 24 hours. So probably most people need about 8 hours sleep And therefore, 6 hours sleep is fairly significant sleep deprivation. So you might be starting to get a sort of a feel for why we're having an epidemic of exhaustion due to just being sleep deprived. Interestingly, this is a very rapid change because 50 years ago, less than 3% of the population slept so little. So humans have evolved over millennia, and we can't possibly expect to cope with a change as drastic as that in terms of sleep duration just over a 50-year period. We sleep different schedules at weekends, and this has been termed social jet lag. So I'm sure you're all familiar with this. Friday night, you tend to stay up later, maybe get up later on a Saturday and a Sunday. And what you're doing then is allowing your sort of body clock to shift a bit later. So one of the reasons why Monday mornings always seems so unpleasant is that when the alarm goes off, although the clock says 7 o'clock, your brain thinks it's 5 o'clock in the morning. So it's screaming at you, why are we getting up in the middle of the night? So this is a kind of really interesting social phenomenon that we're living with at the moment. Interestingly, the more we light up our lives, the less we sleep. So by far the most powerful external signal regulating our rhythmic biology between wake and sleep, eating and fasting is light. So we've all evolved to be out under the sun, and humans aren't great in the night. We haven't got great night vision, so we're not going to be out looking for berries in the night because we won't know which ones are red and which ones are green and so on and so on. So we tended to sort of hide at night. But now, of course, you've got TV and electric lights, so everyone stays up much later. So the more we light up our lives, the less we sleep. The other interesting thing is that lighting used to be very expensive, disproportionately expensive, and the cost of illumination has plummeted over the last 50 to 100 years, just paralleling the rise in obesity and the rise in diabetes and the rise in sleeplessness. So in the USA, it's estimated between 50 and 70 million people suffer adverse health consequences from sleep disorders and sleep deprivation. And these include, as I said, very prevalent major diseases like obesity, type 2 diabetes, and so on. So if that wasn't bad enough, we're in the middle of a very large, very unlicensed experiment in human health, where we have about 1 in 5 people who now work some form of shift. So whether this is earlies, lates, or night shifts. So this is a full frontal assault on your internal biological timing mechanism, and nobody's really paying attention to what happens as a consequence. But if you see, this is just some lay information, higher rates of obesity and ill health in shift workers. So it's not true to say that nobody's aware of the problem, but we don't have any solutions as currently stands. So, hypothetical question, if you're, flying across the Atlantic, would you like your pilot to be drunk? Probably not. The person driving the train, do you want them drunk? Or someone driving an HGV, do you want them drunk? No, probably not. Interestingly, this is a bit of a complicated table, but I've put a red box around the bit to look at, which hopefully will be helpful. So this is basically comparing blood alcohol concentrations of 0.05 and 0.1% in terms of a whole bunch of cognitive reaction speed type of cognitive brain tests. And what these numbers are, these are the number of hours of wakefulness. So this is a study where they just experimentally kept people awake and kept testing them, and they had some drunk people and they tested them as well. So if you're awake for between 17 and 18 hours, it's equivalent mental performance to a blood alcohol concentration of 0.1%, which is significantly above legally intoxicated, so very impaired. So whereas you wouldn't let someone drunk fly the plane, no one ever checks if the pilot has slept. So does it matter? Well, it turns out it does. Look. All of these disasters were caused by sleep-deprived people making a mistake. And it's interesting that of the industries that we talk to, big oil and big gas are by far the most concerned, along with the airlines. And you may sort of see why that might be. So the reputational risk of Exxon Valdez or something similar is enormous, and there are others, numerous others, okay? So, as an example, when workers get flown out to an offshore oil terminal, they all have to have an alcohol and a drug screen, but there's no testing, and there is no test for how tired they are. And that's, of course, something that the companies would love to have an answer to. Those are big disasters, but there's a steady attrition of shift workers dying. And this is just one example. But because they die individually, this unfortunate individual killed a number of people on the way home from a night shift. And this happens all the time. So that's probably not a good thing. What are other consequences of trying to fight against your clock? So we and others have been doing some work looking at night shift as an exposure, and then looking to see what happens to people who've been exposed to night shift. So this is some work where we looked to identify that night shift work is an independent risk factor for the development of asthma, and there are similar studies around rheumatoid arthritis, obesity, diabetes, heart disease, and a whole host of mental illness as well. So, of course, you might think, well, if you've done a night shift, it should be fine, right? Because you'll be tired and you just sleep during the day. So what's the problem? So have you ever tried to sleep at the wrong time of day? So if you've been jet lagged or if you've done a night shift, you'll have experienced yourself it's very difficult. So why might that be? So this is the two-process model for sleep, which includes two drives. One is the sleep pressure, so that's the longer you're awake, then the more tired you are. But there's this other drive here, the body clock drive, or the circadian clock drive to sleep. So how this works is that when you're asleep here, the sleep pressure, process S, is gradually dissipated. So by the time you wake up in the morning, there's no sleep pressure. Then the longer you're awake, then the more the sleep pressure builds until you go to sleep, and then it comes off again. Underlying this, you also have the circadian oscillation, which during sleep gradually prepares you to wake. And here, as the line's going up here, this is driving wakefulness here. As you get into the afternoon and the evening, the circadian or the body clock drive to stay awake starts to fall away, and the gap here between rising sleep pressure and falling awake pressure gets to the critical point, and that drives the onset of sleep. So the problem is if you stay awake to do your night shift, your sleep pressure keeps building, but when you then try and sleep, now you're trying to sleep against this sort of wakefulness drive from the body clock, which is probably why shift workers always sleep for an hour or so less per 24 hours. So what's this fancy picture? So I'd like to take you sort of back in time, if you will. Humans, indeed all life on Earth, entrains its internal clock to the sun, the light of the sun. So when we were moving around by horse and cart or walking, all towns would basically set their time by when the sun reached its zenith in that town. So this is the continental United States, and each of these dials is a different city. And each city had its own time zone, which is fine. Because, you know, if you go on the stagecoach, you'll get there tomorrow. But if you get there tomorrow at 2, 3, well, it doesn't really matter. And who knows anyway. And so they had these quite complicated dials so they could work out what time it was everywhere. And the driver for change was trains, because also you can't possibly run a train system if the time on the timetable is different to the time in the town. So that was the original drive for the imposition of time zones, which are a very artificial construct. So if you live in a time zone, say in the US, or we live in a time zone here, don't we? If you live to the west side of the time zone, your body clock is significantly misaligned or out of alignment with the sun's cycle. And if you live on the west end of time zones, it's a bit like you're doing shift work. And if you look in large enough cohorts of people who live on the west side of time zones have similar risks of similar diseases that befall shift workers. So just indicating how very sensitive we are to time. So this is Oxford. So there's a story about Christchurch, which is that the bells ring at 9:05 in the evening. And of course, the reason for that was that Oxford used to have its own time zone. So we're slightly to the west of London, so our time zone was plus 5 minutes. And the only sort of residual evidence of that is this historic ringing of the bells. So it's not just in the US, it was also here. So I've been talking a bit about timing, and we understand time on the wall. I'm sure we understand the oscillation of the Earth as it rotates, and we see the sun rising and setting. But I have at various times talked about internal time. So internal time is what I'm trying to depict here. So the Earth rotates, here's day, here's night, but in every cell in our body we have a little timing system. And in intact humans, in fact all mammals, we have a central timekeeper here in the brain, which then basically serves as a Greenwich time signal, if you like, to keep all the clocks in our bodies ticking in synchrony. So this is kind of the fundamental basis of all of circadian biology. So what are the output pathways of all these oscillating clocks? Well, there's anything that you can think of is regulated by the clock. And you can look, this is a sort of 24-hour depiction of time. You can see how different sort of human activities or hormonal secretions peak at defined phases of that circadian oscillation. So, of course, my background was endocrinology. We were interested in hormones, and it always used to be a kind of irritating thing that we had to remember the time at which we were taking blood samples because, for example, testosterone's high in the morning and low in the afternoon, so we had to know what time we were taking the samples. And it was only later on I thought that perhaps rather than being a glitch, this could be a feature, and maybe I should take more interest. So how do these cells in our bodies keep time? So it's this mechanism, which I'll go through fairly quickly. You can really regard it as being like a pendulum clock, but in a molecular form. So you get a positive arm, which is 2 proteins, and these drive expression of genes. The genes give rise to proteins, the proteins accumulate, and then these feed back to switch off the activators, rather as a pendulum accelerates and then slows and then goes back the other way. So you can take cells out of a body, you can keep them in a dish, and they'll oscillate forever. So my background's in medicine, and I was talking about this as perhaps being more than a curiosity, but something that might be quite useful. And human inflammatory disease, highly prevalent, and very frequently shows a strong circadian influence. So this is just some data, and this is patients with rheumatoid arthritis, a chronic inflammatory disease of the joint. About 1% of the UK population suffer. And you can see, in terms of symptoms on the left, stiffness, pain, and disability vary massively through the 24-hour cycle. This is so characteristic. It's one of the things we use to make the diagnosis, morning stiffness. It's so characteristic. And it turns out that the driver for that is, and here now we're looking in blood, in the patients is in red, and this is interleukin-6, which is an inflammatory driver. You can see that that is also oscillating, and the peak coincides with the peak of the symptoms. So it's being driven by a clock in the immune system. So we spent some time looking at the lung. Lung's an interesting tissue, inflammatory disease of the lung, hugely prevalent, massive problem, everything from asthma all the way to tuberculosis. The lung is constantly exposed to the external environment. So it's also a complex organ with lots of different types of cells. But if you take these cells out individually, these are the lining cells of the airway. You can see they have a circadian oscillation in a dish. We can take out the professional immune cells. These are macrophages. These also oscillate in a dish. So it's a highly circadian tissue. So we just did some very simple experiments at first, and this is just in a mouse, getting them to inhale some lipopolysaccharide, which is like a bacterial protein kind of stuff. And these cells here are neutrophils, which come into the lung in response to an inflammatory challenge. And I think you can readily see that there's a lot more neutrophils here, and this is CT0, this is dawn, than at any of the other time points. So this is saying that all other things being equal, the response to a pathogen or a mimic of a pathogen varies enormously by time of day. We looked, and a number of the sort of mediators of that inflammatory immune response also showed some changes through time. And this is a particularly important one, which I'll come back to a bit later on. So we naturally thought that if this process in the lung was varying by time of day, if we interfered with that nice molecular machinery, just in those airway lining cells, which are sort of sensing the environment, we should be able to sort of stop that time of day oscillation. And that way we could sort of test whether it was really something coming from inside, from the internal clock, as opposed to it being something else. And that's what we did. So we've just done, the mice are in all other ways completely normal, but they just, in those lining cells that I showed you, which normally oscillate, those now no longer oscillate. You can see the time of day variation in inflammation here, but when we disrupted the clock, the very striking thing is there's far more inflammation at all the time points, and you can see that this is hugely exaggerated at all time points. So it turns out that the timekeeping machinery might be doing something actually quite profound, even beyond telling time to the tissue. I pointed this inflammatory mediator out before. This oscillates in the lung, and its oscillation in the lung coincides with the sort of peak and trough of the inflammatory response. But when we disrupt the timekeeping just in that one cell type, you can see there's a massive increase, and this is the only mediator that shows that increase, which is another fascinating thing. So if you're biology and you're looking at biology through the point of view of evolution, you only need to find a rate-limiting step, put that under the control of the clock, and then the whole pathway becomes under the control of the clock. So it's an extremely efficient and parsimonious way of hooking multiple biological processes onto this ancient timing system. So CXCL5, which is an inflammatory mediator, is very strongly regulated by the clock. So we then wanted to test whether, if you just remove that single inflammatory mediator, what effect does that have on this time of day change in inflammation? So here's a very strong time of day variation in inflammation, roughly 5 times as many neutrophils in the inflammatory response at dawn than at dusk. And just removing a single mediator completely flattens that difference. So again, it just was an elegant proof that the clock is acting through a single gene to regulate the complete orchestration of lung inflammation. And there's some controls over here. So if we were able to target these epithelial cells, these lining cells, which are also called club cells, we may be able to test how well glucocorticoids work. So glucocorticoids are natural hormones, but they're exploited in medicines. About 2 to 3% of the UK population have a regular prescription for a glucocorticoid medicine. They're the standard of care by inhalation for treatment of asthma. So these are very commonly used medicines, and their effect can vary. So some people respond and some people don't. So understanding why some people respond is kind of important. So we thought we'd try to link this all together. And what do we know about the timing and glucocorticoids? So this is a nice study that was done where the investigators here have pulled data out of clinical trials. So these are published clinical trials, some going back to 1964, of the use of various anti-inflammatory drugs. So these are glucocorticoids, prednisone. So what they did was not just to see if taking the drug had a beneficial effect on inflammation, because it does, but they looked to see whether the time of day at which people took the drug made a difference. And all of these 7 trials, with a number of them with glucocorticoids here, revealed a positive signal showing that the time of day made a difference. So that's kind of important because, as I said, we have a significant proportion of the population are regularly taking glucocorticoids, and currently there's no information about what time of day to take them. So can we get any insights using our sort of experimental system? So here we're looking at inflammation, and then we're using dexamethasone, which is a glucocorticoid, which is an anti-inflammatory glucocorticoid. Dexamethasone, of course, recently had a second chance of life because this was the first drug shown to be beneficial in COVID pneumonia. So although it's an old drug, it's still a pretty useful drug. And what do we see here? Well, we can see that inflammation here drives up a lot of these inflammatory mediators, and the dexamethasone inhibits a number of them. Not all of them, but a number of them. When we disrupt the timekeeping mechanism in a single cell type in the lung, you can see that CXCL5, this key one that I've illustrated to you earlier, seems to be the real critical link. Now, this is not inhibited. So here it is in natural wild-type animals, but here, when we've disrupted the timing, it's now not affected. And what do we see when we look at neutrophils, these inflammatory cells pouring into the lung? See that normally dexamethasone inhibits that, but when we've stopped the timer, it stops working. So now we have a molecular explanation for the clinical observation that the time of day makes a big difference. So what about macrophages and lung defense? Macrophages are these professional immune cells. They patrol around our body, and they're the first line of defense against bacterial infection, viruses, and so on. So these are some studies in human macrophages. So what we can do is just take some blood, differentiate the cells into macrophages in a dish. So these cells have been out of a person for about 2 weeks. And then we just give them a signal, which is just a bit of a serum shock, and you can see that their response to an inflammatory challenge shows a strong time of day variation, even though they've been out of a person for a long time. And this is all being driven by their internal timing system. We were the first to identify small molecules which could bind to and regulate the molecular clock, and we were able to show that these molecules here can inhibit the inflammatory mediator interleukin-6, the one I showed you earlier, in a number of these different types of cells. So macrophages, very important for defense, very rhythmic, and can be targeted by drugs. What do? Well, when bacteria are inhaled, the macrophages will attack, digest, and destroy the bacteria. So this is a pneumonia challenge. So pneumococcus pneumoniae, which is a prevalent human pathogen, causes pneumonia. So here we infect a mouse either during the day or during the night, and we see what happens 24 or 48 hours later. So after 24 hours, not much going on. By 48 hours, you can see there's a really big difference in how the mouse has managed to cope with that bacterial infection. This is the lung, and this is in the bloodstream. So the animals infected during the night have got far less bacteria. So this is kind of important, suggesting that depending on the time of day at which you inhale or encounter bacteria, that's going to make a difference days later in how you manage to cope with that infection. So to sort of cut a long story short, we did focus in on the macrophages, and what we were able to show is these are macrophages, and as they eat bacteria, the bacteria fluoresce, so we were able to sort of count them, and either in vivo or ex vivo in a dish, you can see that when we disrupt the timing mechanism in the macrophages, the macrophages actually become superphagocytes. They eat a lot more bacteria. So this is kind of interesting because we're always worrying about antimicrobial resistance. So if you can think of a clever way of harnessing the host defense and just making it better, this could be kind of quite a useful thing to do. I'll just show you this. I hope it works. So these are what macrophages normally look like. So they flatten out, and you can see these big red bands. This is cortical actin, and the flat surface is where it's stuck down onto the glass cover slip. Here, this is what happens when you disrupt the timing mechanism. So the macrophages look completely different. So this is all dependent on timing. And to cut a long story short, we found that the major difference was under resting conditions, the macrophages looked very different. After they'd encountered bacteria, they all look rather the same. The differences are in the red spots. And again, to cut a long story short, we identified the mechanism, and it's all through a particular molecule called Rho-A, which regulates the cytoskeleton of the cell. So that was a direct link between the clock, the cytoskeleton, and the ability of the macrophage to move around. So chronic inflammatory disease is a problem. I mentioned rheumatoid arthritis earlier, and these are patients here just showing again this joint stiffness and interleukin-6. So it's the same thing happens in mouse models, where during the day the pore size is bigger than during the night. So again, this is something, this clock capture of the immune response is seen all the way through evolution. And you can see, again, that compared to the controls, you can see in the red, the inflammatory arthritis, you can see all of these mediators showing dramatic changes through time. And if we drill in a bit further and look here, each vertical line is a different time point, and each horizontal line is a different gene. And you can see here in the control animals, all of these genes are normally highly rhythmic in a joint. And in inflammatory arthritis, they all lose rhythmicity. But interestingly, over here, these genes normally are not rhythmic, and they acquire a rhythm only in disease. Now, if your drug works on a target which is only actually expressed at this time point, you can see why knowing that would be useful, and then designing your drug trial to make sure the drug is given at the right time of day could be really critical. So just to look at all that. I talked about shift work, and I talked about time zones. The other thing we do now is we have dim light in the evening, and this is when we're sort of watching TV and whatnot. And this also is causes a phase shift. So again, we're beset all around us with insults to our timing system. I mentioned earlier shift workers, and these are, again, some more data on shift work, looking at a very prevalent disease that affects the liver, non-alcoholic fatty liver disease, which is when the liver accumulates a lot of fat. It's a risk factor for diabetes. So here we're looking at shift workers, irregular shifts, permanent nights, and this is a measure of liver fat content. And you can see there's a significant increase in liver fat. And the irregular shifts is interesting, because irregular shifts mean that your body can never quite entrain to the new rhythm. So it's probably more disruptive than people who do permanent nights. And you can see the huge numbers of people who this is affecting. So we've done some experimental work on NHS shift workers, and there's a lot of stuff here, but I'm just going to draw your attention to this final panel on the right. Here we're measuring how sensitive people are to the hormone insulin. Insulin is really critical to allow you to cope with meals, and of course, as you If you become resistant to insulin, you get diabetes. So after night shifts, this is insulin sensitivity. You can see this dramatic drop in insulin sensitivity after 3 night shifts. So the effects are very large. So circadian misalignment is very prevalent. Misalignment drives common disease phenotypes in people, and that is also seen in animal models. We'd like to do more in people, but conventional epidemiology has limits because we don't randomize people to do night shift, and people do night shifts for all sorts of reasons. Animal models are useful, but people are complex, and we should be trying to get as close as we can to people. So in the last segment of the talk, I'll just talk about some very recent work where we've tried to use some new statistical approaches to get closer to what's happening in people. So we're trying to look for wider implications of the clock for human health. Now, it's been known for a long time that certain human diseases tend to come in groups, and we call this multimorbidity or multiple long-term conditions. So we set ourselves a view that there must be a shared underlying genetic factor, a sort of cardiometabolic factor, perhaps for non-alcoholic fatty liver disease. I was just talking about type 2 diabetes and coronary artery disease. So these are all common. They're all massive burden on people, families, and the state. So it'd be good to understand a bit more about that. So if we had a common cardiometabolic factor responsible for fatty liver disease, coronary artery disease, and type 2 diabetes, what might it be? This is data on about 500,000 UK residents, and you see we found a number of different chromosomal spots which increase the risk of this common factor. So each of these numbers is a different chromosome across the human genome, and all the significant hits are in colours. We're going to try and do something called Mendelian randomization, so I'll do it quite quickly. But one of the problems if you look in populations and you see that something happens here and something happens there, you'd love it to be a direct cause, but of course there might be something over here which you haven't thought about, which is actually driving both. And there are, of course, lots of famous examples of this. But we can use genetic variation, because you inherit your genes, you don't select them, to see if we can get past this. So we can find genes which are surrogates for the exposure, and that way we're confident that that's the direction of travel. And we can do this on drug targets, because doing a drug trial costs many, many millions, whereas doing one of these drug target Mendelian randomizations just requires data, time, and a computer. So it allows us to go much more quickly. So if we look at drug target Mendelian randomization, this is whole blood gene expression. We find 14 gene targets which are associated with these 3 common prevalent diseases. And one of them, cryptochrome 2, happens to be a core clock factor. It's one of the factors that was in that early diagram that I showed you. We also know that these diseases, the fatty liver disease, diabetes, coronary artery disease, they're also enriched in shift workers where there's been an environmental shake-up of the clock. So it seems like this might be an interesting gene to pursue. What do we know about cryptochrome 2? Well, this is looking at all the genetic associations of cryptochrome 2, and lo and behold, by far the strongest is fasting glucose, which is, of course, a measure of how we're handling carbohydrates, and it also intersects with things like insulin action. So I'm going to go through this fairly quickly, but we start off with the genetic association, which I just talked about. We're able to extract the variants from the genome. We can look at cryptochrome data from outcome genome-wide associations, and then we can do our Mendelian randomization where we look at our genetic variants and we can see whether, if we had a drug targeting cryptochrome 2, what kinds of things might it do. So this is, as it were, a hypothetical drug trial. We haven't got a drug that targets cryptochrome 2, and we haven't done a drug trial. But if we had a drug, this is the prediction of what would happen. First of all, let's look at lifespan, because longevity is quite an important measure. It's relatively easy to tell if people are alive or not, so it's quite a robust one. And you can see that if we lower glucose via targeting cryptochrome 2, we anticipate significant elevations in lifespan. So that's kind of important. Type 2 diabetes is reduced, which fits because that's really how we got here, right? We were concerned about that. But here, now we're looking at atrial fibrillation is also very significantly reduced. The dotted line is no effect. The increase is this side, reduction is this side. Atrial fibrillation is an irregular rhythm of the heart, reduces cardiac output, and massively increases your risk of a stroke. So it's prevalent, more prevalent as you get older. And look, there's an impact there as well. And what about other things? We're looking at the volume here of adipose, how much fat you've got on your body, reduced, and how much fat you have around the midriff, around the abdomen, again, reduced. So, that's the end of my presentation, just to sort of go through some of the points. Hopefully I've interested you in the idea that circadian control systems are extremely ancient. They go all the way back to bacteria, and they underpin essentially all of our biology. Even if we're not that interested in circadian biology is quite interested in us, so we can't really ignore it. Modern life offers a huge and unprecedented challenge to this ancient super system. And it's, of course, as I said, illumination, artificial lights, which allow us to do things in the dark which we'd never have done before. There are major implications for health, health span, and also for medicine. And we probably now need to embed clock logic in drug discovery and in clinical trials, as well as more broadly across healthcare practice. Sleep and circadian science is advancing fast. The Nobel Prize in 2017 for physiology or medicine was awarded for the discovery of that feedback loop, the molecular clock. And it was really for the promise that this may hold for medical benefit. So we haven't actually got to the medical benefit as yet, but hopefully that's the next thing. And hopefully I've also interested you that there are lots of opportunities to live better, longer lives, use existing treatments better, for example, at the right time of day, and to prevent illness by understanding how our biological timing system and sleep work. So obviously I didn't really do this on my own, but I'm extremely grateful for the opportunity to come to Oxford. These are people in my group who've contributed to the work. I've had very generous support from the Wellcome Trust, the Medical Research Council, and the National Institute for Health Research, which is a sort of an applied health research branch of the government, which now funds a better sleep theme within Oxford, but the only one in the UK. So it's very exciting, and there's a lot of pressure on us to now deliver some of the exciting benefits for human health. And of course, I'd really very much like to thank Hugh and Tim and to all of you for the opportunity to talk to you. Thank you.
And we have time for some questions, so please ask away.
Why does a power nap work, a 15-minute power nap?
Sure. So do you remember the slide I showed where the sleep pressure builds the longer you're awake? So if you have a short nap, that dispels some of the sleep pressure. So then it just buys you some more hours until the sleep pressure again gets high enough to drive you to sleep. So if you're aligned with light-dark, and your circadian oscillator and your sleep pressure oscillators are kind of aligned, then as you head into the evening, 10, 11 o'clock, you've got your circadian drive is dropping right away to stay awake, your sleep pressure is rising, and that's when you fall asleep. The power nap in the afternoon after lunch, it just sort of pays off some of the sleep pressure and allows you to have an extra hour or so.
Someone's mentioned something about a whole bunch of little cells in your brain that get really active at that point, astrocytes or something they were called. Is that a thing?
Well, huge numbers of brain cells respond to sleep. It's not just astrocytes, it's virtually all of them. And fascinatingly, one of the purposes of sleep probably is to allow synapses, which are the connections between neurons, to reset. So there's a cost of neuronal transmission. As the signals are electrically going from nerve to nerve, there's a build-up of highly phosphorylated proteins in the nerve terminal, and these take a while to kind of then be kind of cleaned up and processed. Also, when you sleep, the blood-brain barrier opens, and toxins, toxic proteins, particularly things like the beta amyloid, the proteins that build up in Alzheimer's disease, these then pass out from the brain through into the peripheral blood where they're cleared. So it's an amazingly active time for your brain when you're asleep, even though you're not conscious.
Thank you.
I seem to remember that when you put people down in a deep mine and leave them but to choose how long their day is, that it turns out to be 23 hours a day rather than 24 hours a day. Am I wrong? Does this have any effect on...
You are wrong. It's the short answer. No, but let me give you a slightly more considerate... So this is really, really interesting. So we call it a circadian, circa, approximately, DM day. So it's approximately a day. So I don't think that any organism on Earth has an exactly 24-hour circadian period. That is the length of the oscillator when all other external cues are removed. So there were a whole bunch of exciting experiments done. It was actually, I think it was World War II bunkers in Germany where people were sent down to free live. And humans have a circadian period length that's slightly longer than 24 hours. All diurnal animals tend to have an endogenous circadian period of slightly longer than 24 hours. And you know this yourself because if you don't set an alarm, you'll tend to get up a little bit later each day. And if you fly, it's easier to go to the west than the east because our endogenous period length is slightly longer than 24 hours. Interestingly, nocturnal mammals, like mice, their circadian period is slightly shorter. And we respond differently to light. So if we see light, it delays our clock, whereas if a mouse sees light, it advances the clock. And that's probably how the diurnal and nocturnal animals split.
So do different people need different amounts of sleep? And what, in your opinion, is the best time to go to bed and get something awake?
So that's a very easy question. No, so people, so the amount of sleep people need is quite hard to be very precise about, but currently Britain is a sleep-deprived nation, I would say. But yes, like with all traits, if you measure, there's a sort of a distribution, so not everybody needs the same amount of sleep, but people tend to underestimate how much sleep they actually need. So I think that probably you're looking around about 8 hours. Some people need a little less, some people need a little more. It's very interesting if you do the epidemiology. We've been working a lot with huge biobanks, 500,000 people. And there are some odd people. So there are some very short sleepers and some very long sleepers. And the health outcomes for the short sleepers and the long sleepers are equally bad. So it's almost like a U-shaped curve. And of course, there may be confounding, there may be other reasons why people are short sleepers, but it doesn't seem to be a good idea to sort of try and sleep less. The classic one, of course, you may be too young, but Margaret Thatcher supposedly only slept for 4 hours a night, but then I just shrug and say, yeah. It's a huge issue that people think that sleep is a sort of optional, nice to have, and they'll come and see me, and they're sleeping 5 hours a night, and they're leading extremely busy lives, and they're very tired, and they're falling asleep in the afternoon, and they want a fix. There isn't one actually, but because this biology, this clearing of amyloid, and the repair of synapses, we don't have any means of sort of hurrying that along.
You mentioned the timing of taking a drug, and many of us are used to having a prescription from the doctor saying 1 after breakfast, 1 after lunch, 1 in the evening. I mean, when would one get in the future a prescription that says you should take 3 times a dose at a particular time?
So we already.
Is that likely to happen?
Well, so anyone here take statins? What time of day do you take statins? Yeah, so it's kind of already happening. So the drug target for statins is a liver enzyme, which is only expressed overnight. So if you take it in the morning, the drug's gone before the target turns up. So that's why you're always advised to take it evening. So it does already happen to some extent, but that's a very extreme example because the drug target is so powerfully regulated by the clock, and the early statins had short half-life, so they had to be taken at night. I think that the future might be something other than a white pill that you take once a day. So, and the other part of that is, of course, it's the person's own biological clock that we need to know the time of, rather than the clock on the wall. Because as I just said, a lot of us are misaligned to the clock on the wall. So it wouldn't make sense for everyone to take the same drug at 9 a.m. Some people may need to take it at 6 a.m., some people at lunchtime. So I think increasingly we'll have more information on our own biological oscillation. This is already happening. So if you look in the medical devices, well-being devices, I should say, so this is like Apple Watches and so on, there's a huge number of devices being marketed which will track your biological rhythm and your sleep and advise you when to eat, advise you when's the best time for exercise. So this is already happening. And people find benefit. It's possibly a placebo effect to some extent because you have to pay quite a lot for these things. But people are finding some benefit in timing particular activities to their endogenous clock phase. And I think that increasingly, that sort of wearable tech is going to be transformational. Also, as we look with living with long-term conditions, what you can do then is you can have people wearing something on the watch and you can see, step count falls off, and that could trigger an alarm. Someone could give you a call, say, what's happening? You're not walking much, and so on. So I think that increasingly wearable tech is going to put us much more closely in touch with our endogenous timing system. In a similar way, I know people with diabetes who wear kind of continuous glucose sensors. Originally, those were invented with the idea that people would measure the glucose more regularly because they wouldn't have to keep jabbing the finger. But what actually happened is it resulted in a huge improvement in control of diabetes because people could see the response of their body to a meal or to exercise, and because they were getting more information, their brains were able to process this information and they could learn how to live with the diabetes much better. So there are already examples of this.
Thank you. Edith.
I'd be very interested to know if there is a link between the amount of sleep someone has and Alzheimer's disease, because you've mentioned a lot about the clearing of beta amyloid, isn't it?
Yeah.
In Alzheimer's.
So it does appear there is. So there does appear to be a link between sleep duration. Short sleepers are at higher risk of Alzheimer's. There's also some evidence, certainly in experimental models, that circadian disruption, so misalignment, shift work, these kinds of things, can increase the risk of Alzheimer's also. In human studies, of course, it's a lifetime of exposures, so it can be tricky to sort of decipher some of it, but it does appear that there is a link there.
Too scary.
Yeah, and it's really striking. that if you look at what happens if you just experimentally sleep deprive people and measure what happens in the blood, you can see that these beta amyloid proteins are sort of really rising quite steeply. And I say it kind of makes sense, because if you're impairing that sort of physiological repair window, it's not surprising that over time things go wrong.
Are there differences around the world depending on how the day length varies? I mean, obviously in northern climes and southern climes, the day length's very different.
So humans are actually sort of African, really. So we came out of Africa. So the best answer to your question is to look at really sort of northern adapted mammals. And if you look at Arctic mammals, it's really interesting. They've got very weak circadian oscillators, because it's either day or it's night. And so if you look at reindeer, they've got very, very weak circadian oscillators. But animals that from the temperate zones all the way through to the tropics, it's far more predictable that it's going to be this oscillation between day and night. To extend that, of course, where we are in a temperate latitude, we get changes in day length. So that would be a trigger for seasonal animals like sheep for breeding purposes and so on. Humans are probably not particularly seasonal, but humans sleep more in the winter than in the summer.
I wondered if your research tells us anything about diseases with the circadian element, but where the period is not days but months or years, something like cluster headache.
Yeah, so that's interesting. So we're interested in all aspects of rhythmic biology. So I was talking about circadian, where the period length's about 24 hours. There's a whole bunch of stuff which is ultradian. So these are oscillations much shorter than a day. And what you're talking about is infradian, which are oscillations where the period length is much, much longer. So the only real work that's been done on that has been looking at seasonal, circannual changes. And mostly that's been in sort of from the point of view of, seasonal breeding animals, which of course are very important for agriculture and that kind of stuff, because these animals only breed once a year. So interestingly, the infradian rhythms, they still seem to need a circadian oscillator, but there's another mechanism we all have, which is a day length calculator, which is kind of cool. So what happens is when your circadian clock tells you you're approaching time for sleep, a hormone rises in your blood called melatonin, which you may have heard of. It prepares the brain for sleep. So when the melatonin's rising and your retina is expecting to see the light going right down, if the light's still up, the light will then suppress the melatonin, and so you get that discordant signal, and that allows us to detect that this is an abnormally long day length. And that's how seasonal breeders like sheep and so on are able to tell whether it's spring or autumn. But your specific question about rhythmic cluster headaches, I don't think we understand the timing mechanism of that.
We've got time for perhaps. I wonder if you could make a prediction if and when mankind occupies other planets.
Excellent question.
Oh, I'll stop now. Do we impose a 24-hour circadian rhythm even though Mars doesn't have a 24-hour
Exactly.
What happens on the International Space Station?
Exactly right. So these are really, really, really interesting questions. So essentially all organisms with a circadian timing system have a limit of entrainment. So that means that we can sort of move our circadian period a little bit, but we can only probably shift the midpoint of our sleep by about an hour a day, which is why it takes, if you fly over 8 time zones, it takes about 8 days to reacclimate. But that's assuming that the period length is still 24 hours. What you're saying is, if it wasn't 24 hours, what if it was 25 hours or 26 hours? How would we do? So if the difference in day length is small, like an hour, then you can entrain to that. But if you start stretching the day length longer than that, or making it short, significantly shorter, say 20 hours, 10 hours of light, 10 hours of dark, it's impossible to entrain. So what happens is your body tries to follow the short or the long day, does it for about 3 or 4 days, and then there's a sort of huge sort of, waving around, and then it tries again for a few days, and then it waves around again. So you never entrain. And if you do that experimentally, it's extremely deleterious, as you can probably imagine, because none of your internal timing systems are entrained with the external environment. So what I think would have to happen is there would have to be an artificial imposition of 12-12. So we do this, in the Antarctic residential stations, because there's continuous darkness for months, or during the Antarctic winter. On the residential sort of pod, they impose 12-12 light-dark cycles in order to sort of maintain a sort of healthy environment. On one occasion, Greenpeace managed to get some people down there, and they were a bit more anarchic, so they didn't. And the traces, the actigraphy, their sort of activity traces, are all available, and it's a good example of this was really chaotic. They all ran out of time with each other. They were sleeping at odd times, and the duration of sleep wasn't consistent. But it's extremely difficult, because obviously there's no evolutionary mechanism to adapt to a different day length. Of course, what's interesting also is that the Earth's rotation speed is slowing. So day length in dinosaur times was longer than it is now.
How much?
Oh gosh, I don't know. It was a few hours longer, I think. And of course, we can't go back and ask the dinosaurs what it was like, but we anticipate that because the change was very, very slow, that subtle changes in the genetics that underpin that oscillator would just be positively selected for over the millions and millions of years, and there's been a bit of time compensation. But this system is interesting because most of life isn't mammalian, isn't warm-blooded, and most enzymatic reactions go faster when they're warmer. So you might think, well, what if you're a frog? If you're a frog in a hot country, maybe your day is shorter because all the enzymes are working faster. But the circadian clock is temperature compensated, so it's always 24 hours, or approximately 24 hours, irrespective of the temperature. So if you're a bacteria, cyanobacteria, or anything, you can sort of have the bacteria tolerant to a huge range of temperature, but their circadian period length is always about 24 hours.
We've got two people who've been very patiently waiting to ask the question. Lady at the front and gentleman at the back. So if you'd like to ask them sequentially, and then I'll ask David to answer both of them.
I just wonder if you have much sympathy with teenagers and
And at the back.
Yeah, I had a space question, but maybe I'll ask a different one. Do we see things like that in the plant world?
Sure. Teenagers, very, very... This is a really interesting question, and I didn't really have time to talk about it, but for reasons which are not explained, our preference for when we are awake in relation to the solar cycle changes. So our preference is called chronotype. So you've probably heard this, or larks are people who like to be active in the morning, and owls are people who like to be active in the evening, right? So as you go through life, so for example, my baby's very active kind of early in the morning. Teenagers are active, have got a very, very late chronotype. It also differs by sex. So boys have got a later chronotype than girls. So teenage boys have got the latest chronotype in the human population. And after about 21, for the rest of your life, you get earlier and earlier and earlier, which you have this sort of odd thing, like my parents, they're long retired, but they still like to get up about 7:30 and potter around buying newspapers, even though there's no need for them to get up so early. And the consequence for the teenagers is that if you are a teenage boy and you've got a very late chronotype, it's very difficult to get to sleep maybe before about 2 o'clock in the morning. So when you're trying to get up for school, it's like it's the middle of the night. So they miss school, or they turn up and they're half asleep in the first couple of lessons, and this can affect their educational attainment. And if you look at chronotype, which is genetically determined, and you look at late chronotype individuals, their kind of lifespan and their economic opportunities are negatively affected. So there's been discussion about whether, should we have exams starting later in the day, or should school times start later in the day. It's never been looked at systematically, but it's not entirely the fault of the teenager. This is just something in their biology. What's really interesting is why, if you think about a human population, why would you have young people just becoming sexually mature, active at a completely different time to the rest of the population? And the other question was about, it was going to be about space, and then plants, yeah. So this is so if you look at the kingdoms, all kingdoms in life, fungi, plants, animals, have all got circadian timing systems. The principle of the system, the feed forward and the feedback loops, are the same, but there are different genes. And so it looks like it's been independently selected by evolution in fungi, plants, and animals, which is kind of interesting. And the simplest oscillator is in a cyanobacteria, where it's just 3 proteins. And if you just have the 3 proteins, you can synthesize them, put them in a test tube, you add ATP, which is the energy stuff, and these proteins will rhythmically oscillate in the phosphorylation cycle with a period of 24 hours. So you just need these 3 proteins, which is astonishing reductionism. So we don't have the same 3 proteins. But for example, in our clock, we have cryptochrome 2, which I was talking about as this potential drug target for diabetes. Cryptochromes are found in fruit flies. In fruit flies, they're photoreceptors, so they receive light information directly. But of course, a fruit fly, the light can get straight into the brain. So fruit flies have got deep brain photoreceptors. By the time we get to mammals, we've got bony skulls, so the only way light gets in is through the eye. So we've still got cryptochrome, but now it's not a photoreceptor. So you can see that, this is an extremely ancient system. So its kind of fingers are everywhere. And it's right in front of us, but, certainly in medicine, we've tried to ignore it for most of our history.
David, thank you very much for that fascinating lecture.